Hello students, welcome to Newton Gate Academy, where we make your dreams come true. So today's session we are going to discuss about the top 5 MCQs from genetics, that is molecular basis of inheritance. So let's the session begin. So our first question is, a fragment of double-stranded DNA has 38 nitrogenous bases. How many N-glycosidic bonds should be present in this particular DNA? So the options are given. So there are 38 nitrogenous bases. So one nitrogenous base means that there is one N-glycosidic bond is present. So if there are 38 N2 bases, so that means there are 38 N-glycosidic bond present. So the correct option will be 38, option B. So our next question is, which of the following statements are correct about genetic code? So we have all studied about genetic code. There are some postulates. So here 5 statements are given and accordingly 4 options are given. Okay. So first statement is, these are unambiguous. Obviously it is true because there is no confusion in genetic code. Second is, codon are read in mRNA in continuous manner. That is also true because there is no discontinuity in reading this code, genetic code. Third one is, if a nucleotide is deleted or added, whole genetic code read differently. So this is also true. If you change or delete or add the code, the genetic code will read differently. Fourth one is that all amino acids are coded by one codon. No, this is wrong because we all know that any codon can be coded by several different amino acids. Okay. So this last point, one anticodon can pair with more than one codon. That is also true if you take into account Wubbel hypothesis. Because in this hypothesis, it is written that one anticodon can pair with another codon. So according to the given statements, A is correct, B is also correct, C is correct, E is correct. So only option D is not correct. So here option 1, 2, 3 and 5, that means option 4 is correct according to the given question. So our question 3 is, a bacterium has heavy DNA N15 and 15. So if this bacterium is cultured in a medium of N14 light isotope for 3 consecutive generations, then what will be the percentage of light, hybrid and heavy DNA respectively in the 3rd generation? So our options are given. So this is the question. It is coming from the Mendelssohn-Stahl's experiment where semi-conservative model of DNA has been discussed. So according to the semi-conservative model of DNA, one parental strand is there which is again combined with the new strand. So that is the semi-conservative nature. So if that is a E. coli, so this is an E. coli, suppose, which has this heavy strands. Then it is combining with this light isotope N14 and it is giving out 2 hybrid strands. That is semi-conservative replication. This is the 1st generation. So in the 2nd generation, 2 light strands are produced and 2 hybrid strands are produced. So this is the light strand and this is the hybrid strand. And this is 2nd generation. In the 3rd generation, we will get one, two, three. 6 light strand and 2 hybrid strand. That means 75% of the light strand and 25% of the hybrid strand. So the option A is correct. 75% is the light strand and 25% is the hybrid, and no heavy, there is no heavy DNA present, so 0%. That is correct option is A. Fourth question is, here you can see a 5' to 3' polypeptide chain is given. So given sequence when translated must form a polypeptide of how many acids, amino acids. So the options are given. So if we start from this start codon, we have to take a start codon. So AUG we know that is the start codon. So we will first start with this AUG, then again CCU, then UG, UUG, UUA, UGC, and then UUA. So this will be the start codon and UAA is the stop codon we know, right? So we don't consider this or we won't count this. So how many codons we have? 1, 2, 3, 4, 5. So the correct option will be A, 5. Fifth question and the last question of today's session is, if there are 2.4 million base in RNA of eukaryotic gene, how many base pairs in its cDNA? cDNA means complementary DNA. Now you have to understand how this cDNA is formed. We know that if RNA reverse transcriptase enzyme acts upon, then cDNA is formed. So if there is 2.4 million base in RNA, then there will be 2.4 million cDNA will be formed. So the correct option is A. Hope you guys have liked today's session. So see you in the next session. And if you have liked this video, do not forget to share, like and comment in this video. And also subscribe our Newton Gate YouTube channel. Thank you.